Microbial communities, comprising bacteria, fungi, archaea, and protists, are the unseen forces that drive life on Earth. These microscopic organisms thrive in a wide range of environments, from lush forests to extreme contaminated locations. They interact with their surroundings, playing key roles in ecosystems, nutrient cycling, and the breakdown of organic matter. These microbes are essential to Earth's biogeochemical cycles, and they are responsible for many processes that support life, such as decomposition, nitrogen fixation, and even disease control. But perhaps one of their most remarkable abilities is their capacity to degrade toxic synthetic chemicals known as xenobiotics. Xenobiotics are chemical compounds foreign to animal life, things like pesticides, plastics, herbicides, and industrial chemicals. These substances often accumulate in the environment, posing serious risks to ecosystems and human health. But here's the good news. Certain microbes have evolved their ability to break down these harmful compounds. Xenobiotic degrading microbes such as species of Pseudomonas, Bacillus, and Aspergillus possess natural catabolic capabilities, allowing them to metabolize these contaminants and transform them into less harmful substances. These microbes use the toxic elements in xenobiotics such as carbon, nitrogen, and other nutrients as sources of energy to fuel their growth. These microbes have adapted to thrive in contaminated sites, whether it's oil spills, agricultural runoffs, landfills, or industrial waste zones. Their evolved metabolic pathways allow them to survive and perform bioremediation, breaking down complex toxic chemicals into simpler, non-toxic compounds. For example, Pseudomonas and Bacillus are known for their ability to degrade hydrocarbons and pesticides, while Aspergillus, a fungi, has been studied for its role in degrading plastics, both natural and synthetic. These microbes are tailored to break down specific structures in pollutants using specialized enzymes. Microbial biodegradation typically occurs through enzymatic reactions where key enzymes like oxidases, dehalogenases, and oxygenases modify the chemical structure of xenobiotics. These enzymes help microbes break down complex chemicals into simpler, more water-soluble, and less toxic forms. In many cases, these smaller modified molecules can then be used by the microbes as energy sources, fueling their growth and reproduction. Interestingly, the presence of the xenobiotic compound itself can trigger the production of these enzymes. This phenomenon is known as induction. When a xenobiotic compound acts as an inducer, it activates the genes responsible for producing the enzymes needed to degrade the contaminants. As these microbes break down toxic substances, they release essential elements such as carbon, nitrogen, and phosphorus back into the ecosystem. This supports nutrient cycling, which is critical for the growth of plants and other organisms, helping restore soil and water quality. For example, hydrocarbons and pesticides can be degraded into simple compounds like carbon dioxide and water, which are then used by photosynthetic organisms such as plants to produce complex biomolecules. This not only supports the plants, but also benefits the entire food chain, from herbivores to higher trophic levels. Through this process of microbial bioremediation, ecosystems become healthier and more resilient. Microbial activity increases organic matter decomposition, improves soil structure, and enhances soil fertility. Pollutants found in water systems are reduced, improving water quality and supporting biodiversity. However, it's important to note that the benefits of microbial bioremediation extend beyond pollution reduction. By breaking down harmful substances, these microbes help restore ecosystem balance, ensuring that soil and water systems remain fertile and productive, supporting plant growth and sustaining diverse wildlife populations. However, there are challenges to consider. While microbial bioremediation is a sustainable and cost-effective solution, the process can be influenced by environmental factors such as temperature, pH levels, and microbial diversity. Areas with greater microbial diversity tend to be more effective in degrading a broader range of pollutants. In addition, the success of bioremediation can vary depending on the type of pollutant and the microbial species present. Customized approaches may be needed to address specific types of contamination, which is where further research becomes critical. By better understanding the molecular mechanisms behind xenobiotic degradation, scientists can identify specific microbes that could be introduced to enhance the effectiveness of bioremediation in different environments. In conclusion, xenobiotic degrading microbes play a critical role in mitigating the harmful effects of pollutants, reducing toxin compound concentrations, and improving soil and water quality. These microbes not only break down contaminants, but they also contribute to broader environmental health by promoting nutrient cycling and restoring ecosystem balance. The future of environmental restoration lies in harnessing the power of these remarkable microorganisms. As we continue to explore their full potential, we can work together towards cleaner, healthier ecosystems and a more sustainable future for all.